Thank you. So I get the final word. That was not the case when we were growing up. So what I would have given to have had a microphone back when I was, 8 years old. Okay, let's see. Oops. Well, I could tell stories about growing up. So my goal in this talk is actually to bridge what you've heard from the panelists. My passion is taking care of patients with lupus, and my other passion is doing clinical research in lupus. How are we doing with slides? Oh, there we go. Okay. So I needn't tell this audience that lupus is probably the most heterogeneous disease. And several years ago, the Lupus Foundation here was generating a brochure, and they asked advice from me. So I think the way the brochure was initially written, it was like fingerprints, no two lupus patients are alike. And it certainly got the point across, except it sounded a little too criminal. So I said, why don't you change it to snowflakes? So hence the snowflakes. And what I have in this collage are just some of the different manifestations that we see in lupus, from different types of rashes to arthritis to kidney disease, a very challenging disease. So the principles of therapy, very simple. Do good and do no harm. So what I mean by do good is control disease activity. Take someone with active disease and reduce the disease activity. At the same time, prevent flares, because often flares lead to damage. And the name of the game in treatment of lupus is to prevent damage, either from the disease or from the drugs we use to control disease activity. So do no harm, prevent damage from treatment. And the main principle there is make sure the drugs you're using are targeting activity. Don't use toxic drugs for damage that's already occurred, because damage is irreversible. So how do we achieve this? This is how we treat lupus now. It's not a long list of drugs, right? So in yellow are the FDA approved drugs, but in white are non-FDA approved drugs. So we've been using non-FDA approved drugs in lupus for decades. So number 6, immunosuppressives. What that encompasses, that's Imuran, Cellcept, methotrexate. So we sorely need new therapies. We've enhanced the lives of patients with lupus, no question about that. So in this graph, you have on the y-axis, the vertical axis, survival. And on the x-axis, years after diagnosis. So go back to the 1930s and 1940s. A lupus patient did not have a very good outlook. But what came along in the 1940s? Compound E, steroids, cortisone. And if you go on YouTube, you can see some of the first patients who were treated with rheumatoid arthritis with compound E, with steroids. And they go from being bedbound to walking. And then a few years later, patients with lupus were treated with steroids. So you can see from this curve that there was improvement in survival, going from this white line to the yellow line. And then there were additional drugs that were approved, such as the immunosuppressives, Imuran, and so forth. And you see, in fact, better survival. But we're not going to be satisfied until that line is horizontal at 100%. So there are clearly unmet needs in lupus. Patients with lupus nephritis. Now, it obviously depends what metric one uses to measure response. But a really good response occurs only in a minority of patients. Maybe about 30 to 40% of patients with lupus kidney disease will have a very good response with our current regimens. We need better therapies for severe extrarenal disease, to control arthritis, to control rash, and you can go down that whole collage that I showed you. We need to prevent damage. Damage through prevention of flares. We need to get rid of steroids, right? Yes. And it wouldn't be such a bad thing to get rid of the immunosuppressants. And on the wish list is remission induction. And what does that mean? Make the disease go away and stay away. But it's not so easy. So let me just describe the drug development process. The first is a laboratory discovery, and you've heard from our panelists the projects they're working on. The next step beyond that, if one develops a drug, is to do preclinical animal studies to demonstrate that the drug actually is effective, and you heard that we do have animal models for lupus. The other types of studies in animals is to show that the drug is safe. And once that's all been done, then we go into humans. There are different phases of human studies, and I've listed them there. So a phase 1 study would be the first step in humans. And a phase 1 study is generally very small, anywhere from 20 maybe to 70 patients. And the principal goal is to demonstrate safety, but also to get a handle on the metabolism of the drug, what we call PK or pharmacokinetics. And we also want to learn about the biology of the drug, what we call PD or pharmacodynamics. But a phase 1 study is generally small. If all goes well with phase 1, then we move to phase 2. And in phase 2, the questions that are being asked revolve around, is the drug effective? So we're starting to get hints at whether there's efficacy to the drug, but also concentrating on safety. Safety is paramount here. If phase 2 goes well, and phase 2 studies are certainly larger. They can be anywhere from 200 to 400 patients involved. If phase 2 goes well, then the drug is advanced to phase 3. And phase 3, the concentration is on the efficacy, but again, still gathering data about safety. And the FDA currently requires 2 phase 3 studies, 2 pivotal studies for the drug to be approved. And if the drug is approved, then there might be what are called phase 4 studies, which are post-marketing studies to gather yet more information about that drug. Now, I'm not going to quiz you on this slide, but these are some of the principal components of the immune system that are important in lupus. So every one of these has been or will be a target in lupus drug development. So from the laboratory to people. And you heard mention, what happened to my pointer? Well, in this little yellow box, BLISS or BAF. BLISS stands for B lymphocyte stimulator. So studied in the basic science laboratory, but then it became the target of drug development with a drug called belimumab, or you may know it as Benlysta, the brand name. And that took about 10 years from phase 1 to FDA approval. Developing drugs in lupus is very challenging. Why? I mentioned that lupus is the most heterogeneous disease. So how do you study it? Do you focus on kidney disease, or do you focus on arthritis or rash, or do you try to do it all together? But the heterogeneity of lupus makes designing a study very difficult. We don't have many background medicines that we use. We'll use steroids, we'll use immunosuppressants, but I can tell you, steroids, for example, can wreak havoc in trial design. Because how do you know the patient's getting better from your experimental drug, or is it the medicines that they're on? So this is always a very contentious area when we design trials. And we're still wrestling with trial endpoints. You can't just go to the FDA with a truckload of data and say, well, we think the patient got better. You have to have very objective endpoints. And it may sound simple to distinguish a responder to your drug from a non-responder. It's not that easy. But certainly with the belimumab or Benlysta studies, we arrived at a trial endpoint that worked. So that was a big success. But what's amazing, I'm listing the challenges, but right now, there's somewhere around 28 to 30 companies interested in developing drugs in lupus. So that's great. One of the problems we're facing also is in recruitment of patients, and not just a problem in the United States, but worldwide. There are not enough patients to go around. So I've done some math here. It may not be terribly accurate, but I think it gets the point across. So in the New York area, there are probably about 8 to 10 people like myself who are very interested in clinical research in lupus. I'm assuming that maybe of all the patients with lupus in the tri-state area, 10% of the lupus population is cared for by these 8 to 10 investigators. And then not everybody, in fact, only a small percentage of patients are either interested in going into studies or qualify for studies. So if you start multiplying that all across, and you figure maybe at least 300,000 patients in the United States who have lupus, you come up with about 1,500 patients. Well, that's not enough to satisfy what we need for these studies. So right now, I figure there's about a 10,000 patient need to develop all of these different therapies. But despite these obstacles, industry remains very interested. So what's happened in the recent past? We've had some surprising failures. Rituxan or rituximab, we thought was a shoo-in. It was approved for the treatment of lymphoma. It then got approved for the treatment of rheumatoid arthritis. And we've been using it off-label in lupus, and it seems to work. So we designed 2 studies, and both studies failed. We're still using rituximab because we're a believer. And in fact, there's some more studies and more drugs like that in development. So I call that a surprising failure, and it certainly humbled the lupus investigator community, and I must say, we are a tough community to humble. Number 2, successful failures. MMF stands for mycophenolate mofetil, or CellCept. The FDA has a policy about trial design, and if you are using a non-approved comparator, one has to show superiority of your drug, even though you might settle for less. You may settle for equivalence. So this was a study in lupus kidney disease and lupus nephritis, comparing mycophenolate, or CellCept, to Cytoxan, cyclophosphamide. We'd love to get rid of Cytoxan. No one likes being on Cytoxan. We think mycophenolate is safer and equally effective. Well, the FDA did not allow that. We had to show superiority, and we did not show superiority. We showed equivalence. So I call it a successful failure. It did fail. It didn't get FDA approval, but it confirmed what we thought about mycophenolate, that it was a good drug for lupus nephritis. And then finally, we had a success with belimumab. So start to finish was about 10 years. The drug got FDA approval in the United States in March of 2011. It was history-making because it was the first time that a drug for lupus was approved via this traditional route of a randomized controlled trial. And it has lured a lot of companies into this arena. Since they cracked this tough nut, maybe other companies can crack the nut as well. So how can the lupus community help? Well, you've heard this before. Support research, advocate, donate, do both. Participate in research. Well, that's not so easy. So there are different types of research. And not everybody's comfortable receiving an experimental drug. But you may be able to participate in an observational study. We do those all the time. So we may draw blood from you. In fact, the studies you heard from my brother, I drew blood from a couple of my patients. I flew up to Boston, and I played in the lab for a couple of days. So mere donation of blood. but no broken leg and no laceration in my arm. I went home in one piece. So you could donate blood. That can be helpful to a lot of investigators. And then you could try a new medicine. So here's just a list, a partial list of what's in development, and I'm not going to go through all these different drugs, but it's fascinating biology. But notice the line at the bottom, okay? There are more studies than there are patients. So what have we been doing? We've been going to other countries. Not always ideal. We've run into problems in China with bad infections, the same in Africa. And then you have to question the training of the various investigators to fill out all these different instruments that we judge disease activity. So a very difficult challenge. But I'm optimistic. So I think the future, you will see more medicines. We'll have biomarkers. So a biomarker is basically a blood test that helps you determine what the ideal drug is for a patient, or it might tell you how the disease is doing. So it just will give us more objective data. We'll have individualized therapy. So we'll be able to customize treatments for various patients depending what their disease activity is or what their disease manifestations are. And we want that curve, instead of dipping down, we want the survival curve across at 100%. And we also want to stop the blood clots. We want to stop the kidney failure and the dialysis. So I mentioned this when I was in Buenos Aires at the World Lupus Congress giving a talk. It's important to integrate science. There's no question about that. But there's also no question that treating lupus is a real art. So what have trials of new biologic therapies in lupus taught us? They've taught us to be humble. They've taught us to be logical, and a perfect example is the development of belimumab. A lot of logic applied there, advancing our knowledge gleaned from phase 1 to phase 2 and to phase 3. And we persevered. So thank you.